Hi everyone, Brenda Gustin here, and we're in Chapter 3. I'm going to start with Section 3.1. We're going to be talking about prokaryotes, which are the bacteria and the archaea. Frankly, we're going to spend a whole lot of time talking about bacteria and really barely mentioning archaea. Remember, archaea are one of our three domains of classification. So they are prokaryotes. They look like bacteria, but they have some distinct differences. The main reason why we don't talk about them a lot in this course is because we're really kind of taking our understanding of microbiology entwined with the whole idea of disease, health and disease. And it turns out archaea do not cause diseases, so they're not pathogenic archaea. As I say that, one of the things to be aware of when we're talking about the microbial world is almost everything I tell you that's true, there's probably an exception. So if it turns out that we do find some archaea that cause disease, and I kind of read an article that might be possible, it's really going to be the exception rather than the rule. So we're not going to talk very much about archaea at all, except for me to tell you that they're bacteria-like and they don't typically cause disease. And a lot of these are what we would call extremophiles. They grow in extreme environments. So we'll mention those when we talk about those kind of environments. So from now on, I'm going to talk about bacteria. The other thing you want to remember is that there are prerequisites to this course, and one of them is that you had some biology course somewhere. And so most of what you've learned about cells, you've learned about eukaryotic cells. And remember, eukaryotes are plants and animals and protists and fungi, but not the bacteria. And so you should have some feeling for what's the difference between a prokaryotic cell and a eukaryotic cell. And here there's just kind of a general summary of, what do we know in the comparison between the prokaryotes and the eukaryotes. They talk here about the way DNA is packaged. We are not going to talk about that in this course, but we are going to talk about some things about cell walls that are unique to bacteria, that are different from some of the eukaryotic cell walls you learned about. And the big one, remember that when you learned about cells, you learned about eukaryotic cells, you learned a lot of organelles. Well, and remember, by the organelles, I mean things like mitochondria, Golgi, ribosomes, and that kind of thing. Well, there's no organelles in bacteria, in prokaryotes, that have membranes around them. So that gets rid of things like mitochondria and Golgi and although ribosomes are in both prokaryotes and eukaryotes, and we'll talk about those too. So remind yourself about what you know about eukaryotic cells, and we're going to branch off and talk about prokaryotic cells. This gives you just a little bit of background of where we're going. We're going to talk about some of the things on the outside of the cells, and then we're going to talk about the boundary of the cell, and then some things internal to the cell. We're not going to do every topic or every item that's listed in this chapter because it's just, I always have to cut things down a little bit. So the parts that you need to know about are clearly listed on your study guide. And if, for example, S layers, I'm not going to talk about S layers, but I am going to talk about glycocalyx, and glycocalyx is listed on your study guide. So when we're looking at bacterial cells, one of the first things you say is, what do all bacterial cells, what do they absolutely need to survive? And when you look at kind of the simplest structure of a cell, they have to have a cytoplasmic membrane. They have to have a boundary between the inside and the outside of the cell. And we're going to expand a little bit on each of these parts. So all bacterial cells have to have a cytoplasmic membrane, and then they've got to have that kind of fluid on the inside, and that's called cytoplasm, which you guys already know about. Bacteria have ribosomes, just like eukaryotic cells, and we'll talk more about the importance of that. I'm not going to talk about cytoskeleton, but they do need that, and they have at least one chromosome. And we'll expand on all this. Here it says the majority have a cell wall and a surface coating called the glycocalyx. And so some of you might have that cytoplasmic membrane, or also called a cell membrane, confused with the cell wall. So it's time to get that straightened out because they're also present in many eukaryotic cells. So we're going to kind of expand on all that. This is the picture that we're going to be looking at, and it's got a lot of details, tells you about these different parts, and you can pick out the ones that we're going to be doing in this unit. Now in section 3.1, I'm pretty much asking you to recognize the structures, and you can see the list of the names of structures that you're going to need to recognize in a diagram like this, like the cytoplasmic membrane, which is going to be right outside of the cytoplasm. Here they're showing you, here's a bacterial chromosome right here. This whole purpley thing. Ribosomes, there's lots of these little dots are ribosomes, and the cytoplasm is the fluid inside the cell. Other things we're going to look at, the cell wall. We're going to expand on glycocalyx. We're going to look at and talk about some inclusions, plasmids and flagella, and endospores. So most of these things we're going to be doing, you should be able to find these on a diagram. So before we get into detail on those, let's just talk quickly about bacterial shapes and arrangements. And so remember, bacteria are single-celled organisms, but they don't always just exist as a single cell. Sometimes they're attached to other cells. But a single cell can survive, and they do very well. A single cell carries on all the activities of life: reproduction, metabolism, nutrient processing, etc. But sometimes they act as groups when they're attached to other cells or in mixed communities, and we'll talk a little bit more about that. One of the things that's always good to know is that bacterial cells are about 1 micrometer in size, and so if you were going to look, I think what the common number is 0.1 millimeter is on the edge of what we can see. So 0.1 millimeter is 100 micrometers. So you need 100 bacteria to even possibly be able to see them without a microscope. So that's a good number to get into your mind. About 1 micrometer is about the size of a bacterial cell. And it goes on about here. Here's some bacteria that are really big, and you can see that they're almost 200 micrometers, or 150 micrometers across. And so this would be the exception, and that goes into that category of everything I tell you about bacteria, there's always going to be an exception. So if you kind of start with one micrometer for bacteria, that might give you a perspective, because eukaryotic cells are going to be bigger, viruses are going to be smaller. So let's look at the 3 major bacterial shapes. And I'm going to give you what I say are 3 shapes. We have a coccus, a bacillus, and then a spiral. Now in this diagram, it also has a vibrio, which you can see are kind of curved. I kind of going to group those in with spirals. Okay, so I'll just say, if I ask you, what are the 3 major shapes of bacteria? They'll say coccus, bacillus. Some people call them rod-shaped. And you can call it spirillum or spirals. And notice, again, there's a couple different kinds of spirals here. So I'm going to lump all these together in spirals. And then this branching filaments, which I'm not going to go into. Coccus-shaped bacteria mean that they're round. So here's lots of round bacteria. They're coccus-shaped. And it's telling you, oh, they may or may not have a little point on the end, but generally you're looking for bacteria that are round, for coccus-shaped. Bacillus-shaped, also called rods, are rod-shaped. They're longer than they are wide. So here's some classic bacillus-shaped bacteria. And then this group right here, you notice they're kind of bacillus-shaped, but they've all got some kind of curve going on. So I'm going to just lump all these together, and I'm going to call them spirals. And you can see that there's a difference between a vibrio, a spirillum, and a spirochete, but it's not necessary for our understanding. Just know that when you look at them under the microscope, if they're kind of curved, and see how those all have kind of a curve in them, and these are typically straighter. And so all these, I would classify them as spirals. And there's always some variations too. Okay. The other thing is if we have three major shapes of bacteria, Bacillus, Coccus, Spiral, sometimes they're in certain arrangements, which means they're associated in certain ways with other bacteria of their same shape. And so we call that the bacterial arrangement. And in particular, coccus-shaped bacteria are noted for making different arrangements. So some of the vocabulary you should be familiar with are, here you have, and it's going to depend on how the bacteria divide. But as they're growing, they're going to divide, and you can see they're dividing in one plane. So one cell divides into two. And ultimately, some bacteria will just stay in these pairs. And so because they're round and there's two, we call them diplococci. And looking at certain kinds of bacteria under the microscope, you'll notice that, if they're making diplococci, it's a hint as to what kind of bacteria it is. Some bacteria will end up in round cells in chains, and so we call them streptococcus or streptococci. Coccus is singular, cocci are plural. So streptococcus means round cells in chains. So if somebody has strep throat, it got the name from the arrangement of the bacteria, from streptococcus. In fact, for strep throat, streptococcus is the genus name. Here's another way that some bacteria divide, some coccus-shaped bacteria. If they divide into two planes like this, they can end up in groups of 4, and we call those tetrads. So tetra, 4, and so 4 coccus-shaped bacteria is called a tetrad. Another common shape I want you to know, or a common arrangement, is if we get certain cells will divide in many different ways, and then they end up in these clusters. And so the clusters, when it's like that, we say it's called staphylococcus. And so chains of single round cells, we'll just say it's a coccus. A pair of round cells is a diplococcus. Chains or strips of round cells are streptococcus. I think strepto sounds a little bit like a strip. Groups of 4 coccus we call tetrads. And then round cells in clusters, we call them staphylococcus. And so just like streptococcus, staphylococcus can be a shape or it can be a genus. So bacteria that cause staph infections are in the genus Staphylococcus. And you can look under the microscope, and if you see round cells in clusters, it's a pretty good hint you've got Staphylococcus. Okay. Here it tells you rods are less varied in their arrangement because they only divide in one way, but you can find some in pairs, so they might be diplobacilli, and you might find them in chains called streptobacilli. Usually they're in singles, and we just call them bacillus. That's the last thing I have for this. So in this section 1, 3.1, we're just looking for some of the structures, and we're going to expand on some of these in the next. We're just looking for the structures and the diagrams, and we'll expand on these in the next section.